So here is our second video for chapter 24 on early life on Earth. So we're going to start off today just going over bacteria and how they respond to things, which is kind of weird. You guys know, like, you have a brain, you respond accordingly to whatever stimulus is presented to you. It might be a good response, it might be a bad response, but it is a response nonetheless. They found that bacteria, they're just single cellular organisms. They don't have like a brain and organs and all these things. They're very simple. But they do show something called chemotaxis. And what that is if you give that bacteria a stimulus, it'll either move toward it or away from it. Now, what determines if it moves toward or away, we don't know. But nonetheless, it does move and respond in one way or another. So, with respect to movement, how bacteria move is by the back of it. It has like this whip-like structure called a flagella. It's pretty much just like a long thing on the back of it. I could draw it for you guys real quick here. So if we have our bacteria cell, we'll just do a bacilli here because that's the easiest one for me to draw. Here's our rod bacteria. And here would be the flagella on the back of it. So this thing will whip around each way, whatever way it wants to go, for it to be able to move the bacteria. Now, they found that in bacteria, archaea, and eukaryotes, some of these cells actually have a flagella, even eukaryotes, which is kind of weird, but there are single cellular eukaryotic cells. Now, they found that the flagella is made up of completely different proteins in bacteria, archaea, and flagella. What this tells us is, since they are completely different in their chemical makeup, it tells us that they evolved independently of one another. Now, did they start from one single organism and then go through speciation? Or did they all start as different life forms and evolve independently? We're not sure. But regardless, at one time they did evolve independently of one another. We don't know if that's from a common ancestor or if they all started off as different organisms from the beginning. Here is a picture of the internal structure of a flagella. And you guys can see there it has some motor proteins, and those motor proteins help it to move around. So here's just, guys, we have a motor that helps it to spin, and there's some different parts of the flagella as well in order for the bacteria to be mobile. Okay. So the flagella, they say here, likely evolved as existing proteins were added to an ancestral secretory system. Now, with this, guys, when we're talking about that, it probably, they were added, and it was a slow process where these proteins were added to the back of the bacteria. Why, we don't know. But going from a few chapters ago, this is an example of an exaptation because it wasn't used for that. We weren't thinking, okay, let's make this big flagella. Instead, we were just throwing the proteins back there. It's like, okay, let's just leave them back here. And then it's like, we got this big structure. Oh, hey, look what I can do. I can move all around with this. So it wasn't intended for that purpose. Okay, for prokaryotic cells, they don't have a lot of compartments. In other words, we have all these organelles and different things that are for specific functions. Prokaryotes don't have these, but they still got to go through some metabolic functions. So what they will do, guys, is they will actually have like infoldings on the plasma membrane, on their cell membrane. So if we wanted to look, we could have like a cell wall here. Do our flagella on the back. Now on the inside, I'll draw it in blue. The cell membrane might look something like this instead of how we draw it, where it's like pretty much the same or parallel to our cell wall. But what it'll have is it'll have some little infoldings to increase surface area. This way, we can do some metabolic processes using our cell membrane, and we can do things like make ATP and stuff like that, go through the electron transport chain. So the bacterial cell is still able to make energy, make ATP. Okay, different parts of our prokaryotic cell. You guys probably can recall from the past that bacteria has one chromosome and it is circular. They also have this area in the middle called the nucleoid region. This is where our DNA kind of just hangs out. Remember, prokaryotic cells like bacteria do not have a nucleus, right? So they're going to have the DNA kind of just chilling there in this nucleoid region. Along with this, the bacteria might actually, and I'm talking about in addition to that one chromosome, they might have little smaller things of DNA called plasmids. So here's the nucleoid region here. Big DNA right there in the middle. There are our smaller plasmids on the side. Here's another picture here. You guys can see the plasmids are much smaller. All right, and here's the one under a microscope, so the non-animation one. So what these plasmids can do, guys, is we can use them for our benefit, actually, by introducing some different genes into that plasmid and having that plasmid reproduce many, many, many times, and then we have a bunch of DNA with that gene of interest in there. So what's nice about this, guys, is we can actually use these plasmids as antibiotics like insulin and things like that in order to help us get medicine for any condition we might have. All right, so 4 different types of prokaryotes. We have phototrophs, we have chemotrophs, we have autotrophs, and we have heterotrophs. And I know in the past we already went through autotrophs and we went through heterotrophs. We already went through those. So 2 new ones for us here. Phototrophs obtain energy from light. Chemotrophs obtain energy from chemicals. So these are very general terms, and we can get more specific in talking about autotrophs and heterotrophs. Remember, autotrophs are probably going to go through photosynthesis most of the time in order to generate food for themselves. So I'm going to use the chart and go over these 4 terms, guys. Photoautotrophs, they're going to use light as our energy source, along with photoheterotrophs. So they're going to be the same. Difference here, photoautotrophs, their carbon source is going to be carbon dioxide. Now, a photoheterotroph is going to get its carbon source from organic molecules. So it's not going to use carbon dioxide for the carbon source. Instead, it's going to use other organic compounds. A chemoautotroph. So chemo, it's not going to use light, and instead it's going to use organic chemicals instead of the light, but it's still going to need carbon dioxide. Okay, so anytime you guys see autotroph, it's going to use carbon dioxide for its carbon source. Heterotrophs, they are going to use organic compounds for their carbon source. Okay, so when we talk obligate, and I know we went over this definition in the past, obligate means you are obligated to do one thing. So by with the first one here, obligate aerobes. These are going to require oxygen for cellular respiration. If they do not have oxygen, they are obligated to have oxygen. So if they do not have oxygen, they cannot make ATP, and they will die. Okay, that's pretty much it for them. So anytime you guys see aerobic or anything like that, you know it means requiring oxygen. Anaerobic is going to be the opposite. They don't require oxygen. So if it is an obligate anaerobe, it definitely does not want to take oxygen in. Actually, they will be poisoned by oxygen. So definitely not good there. Now, when we're not talking about obligate, the opposite of that can be facultative. So This means that it can survive with or without oxygen. So a facultative anaerobe, if oxygen is there, cool. If it's not there, cool. All right. Anaerobic is going to be, obviously without oxygen. But if oxygen there, it's not a big deal. It's not going to die because of oxygen. Okay, so for nitrogen in our bodies, we know that we need nitrogen for a few things. Amino acids, because we have the amino group in there, and the amino group has nitrogen in it. We also need nitrogen for nucleic acids. So when we are using our cells to make new cells, we are going to need some amino acids to copy that DNA. So how do we make these amino acids? Well, we bring in nitrogen for that. So for this, guys, we have to be, whether we're prokaryotic or eukaryotic, we have to be able to fix nitrogen. Now, we talked about early Earth, and early Earth had nitrogen and ammonia, and both of these were two different gases that contain nitrogen in it. So they were able to use these two gases and start to make nucleic acids with them. All right, next thing, guys, heterocysts. These are thick-walled cells, and all they do is specialize in...
Please call Peanut by 301.
Sorry, guys.
By 301.
So, heterocysts. These are thick-walled cells, and they specialize in nitrogen fixation in cyanobacteria. So if you remember, we went over cyanobacteria. That was the early bacterial cells that were able to photosynthesize, but this is what allowed them to fix nitrogen. They're not photosynthetic, though. So the cyanobacteria is photosynthetic, but the heterocysts are not. We can also find these in algae and fungi cells as well. Fungi, you may look like a plant. It's actually not a plant, and it can't photosynthesize. So, all right. So reproduction. Prokaryotes, they can reproduce like super quickly. For ourselves, it takes a while. We are not going to reproduce fast at all. Our skin cells, we lose our top layer of skin, which is dead, every 30 days. So it takes us a while to reproduce. But bacteria, they divide every 1 to 3 hours, which is why bacteria are so plentiful. Now, why they're able to do this is because, one, they're super tiny. They reproduce by binary fission, which, if you remember, all we're doing there is copying our DNA and pretty much making a new cell wall and cell membrane, just like growing it so we could split it in half. And that's pretty much it. We don't have to worry about copying our organelles and all that other stuff. Since it has one chromosome, too, we don't have to worry too much about making sure the DNA is all good. Okay. So next slide, guys. This is just a summary of everything we went over, but I'm just going to concentrate on the second one here. Prokaryotic diversification can be viewed as the first great wave of adaptive radiation in the evolutionary history of life. Remember from last chapter, adaptive radiation, might have been the chapter before actually, we are setting up an ecosystem. We are setting up an environment in a certain area. And with adaptive radiation, what we are able to do is fill the holes in that environment. So it's pretty much we are giving rise to new species because that environment needs this species to make sure every niche is filled in that environment. And with this, guys, with adaptive radiation, later on we could even create, if needed, greater and more complex organisms based on ones we already made. Okay, so prokaryotes, guys, we've been going over bacteria. Now, you guys are like, okay, single cellular, very simple cells, right, not a big deal. But they actually have a lot of genetic variation because, well, they've been evolving for 3.5 billion years. They've been around so long. we are going to talk about other ways that bacteria can change their DNA, because it has changed a lot. Obviously, in 3.5 billion years, yeah, it's changed a good bit. But the primary source of variation in prokaryotes is mutation. Given that most of the time, prokaryotic binary fission reproduction is not sexual, we're not going to have a lot of genetic diversity, because we are not having DNA from two different organisms come together. Instead, we just have asexual reproduction, one going to one. All right, so mutation is our primary source. However, there are some different ones that we will go over here. I'm going to skip this diagram. We don't really need that one. Okay, so our three other ways that our prokaryotic cells can change or evolve DNA. There's transformation, there's transduction, and then there are conjugation. So let's start off with transformation and transduction. Transformation, how this occurs is that prokaryotic or bacteria cell is taking up DNA from the surrounding environment. Given that DNA is going to be foreign for that bacteria, so in taking that DNA and making it part of its DNA, it's going to change things up a little bit. Transduction, our second one, is going to be when that bacteria cell, when that prokaryotic cell is going to get DNA from a bacteriophage. And if you remember, a bacteriophage is a virus that infects bacteria. So when that virus comes in, most of the time, yeah, the virus changes the bacteria into a bacteria reproducing machine, a virus reproducing machine, and overall the bacteria will die in the end. But if it doesn't die right away, we still have some different genes in there, and that bacteria can reproduce. And here is a picture of transduction. Our last one is going to be conjugation. So this is where we are going to use that pili in order to take one bacteria cell and transfer some DNA to another bacteria cell. And here's a picture of it. And we are going to transfer some DNA in that process, and that will change the DNA of that bacteria cell that is receiving the DNA. Now, when we're talking about conjugation, we are going to have two different terms for those two bacteria cells participating in conjugation. We call these the DNA that is transferred, it's an F factor. So they can either be F plus or F minus. The cell that is going to give the DNA, we call that one the F positive one. The one that is going to receive it, that is going to be the F negative one. And here's a diagram of everything that happens during conjugation with one DNA going to another. And you guys can see part of that plasmid is going to from one DNA to the other. Again, it goes from the F positive to the F negative. Okay. All right. So once the F negative cell receives it, it actually is probably going to become F positive, and it's going to look for another cell to donate that plasmid to. So this will, in time, slowly change the whole population of bacteria cells if it is a plasmid that is beneficial to the survival and reproduction of that bacteria. Okay. So our plasmids, this is another type of plasmid. This is going to be antibiotic resistant. So this is a really good plasma for bacteria to have. For us, not so much, especially if it's a bad bacteria. But if the plasmid is transferred over to other bacteria, and if it is an R plasmid, that's going to give the other cells this R plasmid as well. Now, for antibiotics, guys, what's going to happen is when you are taking this antibiotic for some kind of bad bacteria in your body, you are going to be taking some kind of pills to try to get rid of the bacteria. Now, I know doctors will give you, let's say, like, take this pill for 2 weeks, twice a day, and then you'll be good. And then on the pill container, it'll say, make sure to take all your pills. This is very, very, very important, because what you're doing is you are showing the bacteria what the antibiotic pills are containing to make the bacteria die, essentially. If you don't kill all the bacteria, you pretty much just showed them what our defense is against them. So if it mutates in some way, we can get antibiotic resistance. We want to make sure all those bacteria are dead. And that's why we take all of our pills. Okay, guys, so let's stop here for today. We will finish the chapter next time, and then we will all be done with chapter 4. Have a good rest of your day.